Spectrophotometry is a core method in laboratory testing. The principle of spectrophotometry is the measurement of light within a narrow wavelength range, with the wavelength selected by prisms, gratings, or filters. A chemical reaction produces a substance that absorbs light. Measurements can be made in the visible range, which is most common, or in the ultraviolet and infrared regions. Under ideal conditions, the amount of light absorbed by a solution, when illuminated with light of the proper wavelength, is directly proportional to the concentration of its colored component. This principle is known as Beer's law. Importantly, the same principle applies across many analytical methods that measure radiation, such as gamma counters. Beer's law states that if measurement is made in absorbance and plotted on linear graph paper, the concentration of the substance is directly proportional to the amount of light absorbed. In practice, Beer's law is used to calculate concentration by rearranging the formula. A simple application involves comparing the absorbance of a known standard with that of an unknown patient or control sample. However, deviations from Beer's law may occur. These deviations can result from simultaneous absorption at multiple wavelengths, from interfering substances that absorb light, or from stray light transmission. A spectrophotometer has several components. The light source may be a tungsten lamp for visible range measurements, or a hydrogen discharge or deuterium lamp for ultraviolet range. The monochromator isolates a specific wavelength and may consist of a filter, a diffraction grating that works from near ultraviolet to near infrared, or a prism, quartz for visible and ultraviolet light, and glass for visible light only. The cuvette, which holds the sample, may be glass, quartz, or plastic. The detector is often a photomultiplier tube, and the measuring device records the results. Standardization of spectrophotometers ensures accuracy. Wavelength calibration may be checked with hydrogen or deuterium lamps, holmium oxide or didymium filters, or solutions of stable chromogens. These confirm whether the instrument's wavelength accuracy has changed. Stray light can be detected using filters or solutions with sharp cutoff wavelengths, such as NSQ40. Corrections involve changing the light source, verifying wavelength calibration, sealing light leaks, realigning optical components, or cleaning the instrument's surfaces. Finally, there is bichromatic analysis. This is an automated procedure in which absorbance is measured at two different wavelengths. It helps minimize background interference, such as that caused by hemolysis or icterus, by essentially providing a blank for each specimen. Let's review several important analytical methods: fluorometry, turbidimetry, nephelometry, and osmometry. The principle of fluorometry is based on energy emission. When certain compounds absorb electromagnetic radiation, they become excited and then return to an energy level that is slightly lower than their original state. The energy they release is less than the energy absorbed, which means that the emitted light has a longer wavelength than the absorbed light. Fluorometry is extremely sensitive, up to 1,000 times more sensitive than colorimetric methods. However, it has a disadvantage: quenching interference. Quenching occurs when the excited molecule interacts with another substance in the solution and loses some of its energy instead of emitting it as light. The principle of turbidimetry is straightforward. It measures the amount of light that is blocked by particulate matter as light passes through a cuvette. The measurement is usually made with a colorimeter or spectrophotometer. Sources of error include differences between the particle size in standards and in samples, and the possibility that particles may settle out of suspension while measurements are being taken. Nephelometry works a little differently. Instead of measuring how much light is blocked, it measures the light that is scattered by small particles at right angles to the incident beam of light in the cuvette. The amount of scatter is directly related to the number and size of particles. Nephelometry is generally more precise than turbidimetry. Clinically, it is widely used to measure antigen-antibody complexes, such as immunoglobulins IgG, IgA, and IgM, as well as C-reactive protein, rheumatoid factor, complement components C3 and C4, haptoglobin, and others. This method provides high specificity. Osmometry is the measurement of osmolality, which is the total number of dissolved particles in a solution. The molecular weight, size, density, or type of particle does not matter. Only the number of particles counts. Any dissolved substance will change the solvent's physical properties, called colligative properties. These include depressing the freezing point, elevating the boiling point, decreasing vapor pressure, and increasing osmotic pressure. The most practical methods for measuring osmolality are freezing point depression, which is the most common method, and vapor pressure depression. A significant difference between results obtained by these methods can indicate the presence of a volatile solute such as alcohol. For example, osmolality is increased in the presence of ethanol, methanol, or glycol when measured by freezing point depression, but vapor pressure methods do not detect these volatiles and therefore give lower values. Sources of error in osmometry include improper cooling bath temperatures that are too cool or too warm, and particulate matter in the sample preventing sufficient supercooling before freezing occurs. Chromatography is a powerful analytical technique used to separate mixtures into their individual components based on differences in physical or chemical properties. The principle of chromatography is that compounds in a mixture will move differently through a medium depending on their physical characteristics, such as solubility, charge, size, or volatility. There are several major types of chromatography. Liquid-liquid chromatography separates substances based on differences in solubility between two immiscible liquid phases, one aqueous and the other organic. Ion exchange chromatography separates molecules according to their size, weight, and charge. Ions with greater charge densities are held most strongly by the ion exchange material. Gas-liquid chromatography and gas chromatography depend on sample volatility and the rate of diffusion into the liquid phase or inert carrier gas. Retention time is used to identify volatile compounds such as methanol, isopropyl alcohol, organic acids in urine, drugs, and catecholamines. This is the best method for measuring blood alcohol. For confirmatory testing of regulated drugs of abuse, gas chromatography must be combined with mass spectrometry. Thin-layer chromatography separates substances according to their rate of diffusion and solubility in the solvent as they migrate across a medium. Thin-layer chromatography is commonly used to identify drugs, lipids, carbohydrates, and amino acids. A measurement called the R-F value is calculated as the distance traveled by the substance divided by the distance traveled by the solvent. High-performance liquid chromatography, or HPLC, uses aqueous or organic solutions pumped under high pressure through columns. This provides very high resolution with rapid and accurate quantitation. Finally, when troubleshooting chromatography, several issues can arise. A noisy baseline may be due to bubbles, particulate matter in the flow cell, or a leaking filter. A drifting baseline can result from an overloaded column or contamination in the solvent reservoir. Loss of column resolution may occur when too much sample is loaded or when the column packing becomes degraded. Electrophoresis is a laboratory technique used to physically separate proteins and other molecules based on their ionic charge and molecular size. The principle is that when molecules are placed in an electrical field, they migrate depending on their charge. The direction and distance of movement depend on the molecule's isoelectric point and the pH of the buffer. The rate of migration is further influenced by molecular weight and size, the ionic strength of the buffer, and the type of support medium used. There are several types of electrophoresis. These include paper electrophoresis, agarose gel, cellulose acetate, which is the most common, polyacrylamide gel, starch gel, and isoelectric focusing. Each medium has different properties that influence separation and resolution. Electrophoresis is widely applied to separate many biological components. These include amino acids, serum proteins, lipoproteins, glycoproteins, nucleic acids, hemoglobins, isoenzymes such as creatine kinase and alkaline phosphatase, and immunoglobulins. In addition, specific antigens can be identified using immunological electrophoretic techniques, such as rocket immunoelectrophoresis or immunofixation. In summary, electrophoresis is a versatile technique that separates molecules by charge and size, using different media and methods, and plays an essential role in protein, nucleic acid, and immunological analysis. When performing electrophoresis, several problems can occur that lead to errors in the results. If there is no migration at all, the most likely reasons are that the instrument is not connected, the buffer has the wrong pH, or the electrodes are connected backwards. If the bands appear too large or too small, the problem is usually due to overloading or underloading the sample. If migration is very slow, it may be because the molecules have a high molecular weight, a low charge, the ionic strength of the buffer is too high, or the voltage applied is too low. Sometimes the sample precipitates in the support medium. This usually happens if the pH is too high or too low, or if the voltage applied is too high. When you see split or broken bands, the cause is often excessive pressure applied when using wire applicators. Finally, crescent-shaped bands are typically the result of bent applicators or overloading the sample. In short, most electrophoresis errors come from equipment setup, sample preparation, or improper application techniques. Recognizing these issues allows us to troubleshoot quickly and ensure accurate results. Electrochemistry in the laboratory includes three important methods: potentiometry, amperometry, and coulometry. Potentiometry is the basis of pH meters and ion-selective electrodes. It works by measuring the potential or voltage between two electrodes when no current is flowing. The difference in voltage occurs in proportion to the ionic activity of the solution. The reference electrode is usually either a saturated calomel electrode, which contains mercury chloride and potassium chloride, or a silver-silver chloride electrode. The indicator electrode is designed to interact only with the analyte of interest. Examples include the glass pH electrode, which is sensitive to hydrogen ions; sodium electrodes with glass sensitive to sodium ions; potassium electrodes that use a liquid ionophore called valinomycin; gas-sensing electrodes with membranes permeable to gases, such as the carbon dioxide electrode. This CO2 electrode is a pH glass electrode covered by a silicone rubber membrane permeable to CO2. Enzyme electrodes, which are coated with immobilized enzymes that catalyze reactions measured by the electrode. Amperometry involves measuring the current that flows through an electrochemical cell while a constant potential is applied to the electrodes. An important application is the measurement of blood oxygen using the PO2 electrode. In this electrode, the cathode is a metal such as platinum, and the anode is silver/silver chloride. The current observed is directly proportional to the oxygen concentration in solution. Coulometry is a type of titration in which the titrant is generated electrochemically. For example, silver ions can be produced by electrolysis from a silver wire anode. These silver ions then react with chloride ions in the sample to form a complex. An indicator electrode detects the point at which excess silver ions appear, marking the end of the titration. The length of time it takes to reach this endpoint is directly proportional to the chloride concentration, since the current is kept constant throughout the process. In summary, potentiometry measures voltage to assess ionic activity. Amperometry measures current to quantify analytes such as oxygen, and coulometry uses electrochemically generated titrants to measure ions like chloride. Let's summarize the main instrumentation principles in laboratory science. Spectrophotometry measures light within a narrow wavelength range. Bichromatic analysis measures absorbance at two different wavelengths. Fluorometry measures light emitted from compounds that absorb electromagnetic radiation and release energy at a longer wavelength. Turbidimetry detects how much light is blocked by particles as it passes through a sample. Nephelometry measures the light scattered by small particles at right angles to the incoming beam. Osmometry measures the total number of dissolved particles in a solution, using colligative properties like freezing point depression or vapor pressure changes. Chromatography separates mixtures into components based on physical and chemical differences. Electrophoresis separates molecules based on ionic charge and size. Potentiometry measures voltage differences between reference and indicator electrodes, with no current flowing. Coulometry involves titration using an electrochemically generated titrant, such as silver ions for chloride measurement. Amperometry measures current flowing through a cell when a constant potential is applied, such as in oxygen electrodes. In summary, each technique uses light, particles, charge, or electrical properties to detect and quantify substances, all essential for modern diagnostics.